you're starting with the first strand and then you're replicating it, correct?
Depends on the question. Yeah, depends on the question. Yeah. So if you want me to do that, we can do that. Are we ready? Oh, thank you so much. All right.
When you say two strands.
DNA is always double-stranded. The question was, okay, do all DNAs have a start codon and a stop codon? The answer is yes. Okay. You have DNA here and a DNA here, right? Two strands of DNA. Both of these strands, they have genes, right? They have nucleotides. These are your genetic code. Three-letter words are genetic codes, okay? But this gene starts right from here. When you transcribe it, you make your mRNA, okay? Become exactly. This is your sense DNA. Sense DNA. So this is your start codon right here. Start. And at the end, UAG is your stop codon. So every gene has a start codon. And how about the antisense strand right here? Antisense. I have to tell you. I have to. No, not really. Yeah, because both can be transcribed. This is gene, but this is start codon is somewhere else. It's not cannot be opposite of that. So let's suppose. Let me give you an example. If this is for this DNA makes gelatinase, this DNA makes amylase, and the bacteria has gelatin but no starch. This DNA will be sense, and that will be antisense. Now you take the bacteria, put it in starch. This one will become sense, and this one will become antisense. Absolutely. These are code, exactly. Yes, exactly. Same thing, but different things. Different things. So your question, both strands, yes, both strands have start and stop codon, but at different location. Because the length of the gene, some proteins may have 3 million amino acids, other may have only 1 amino acid. I mean, not 1 amino acid, 1 million amino acids. So, just Jackie.
So, for instance, this, like you were showing us the other day, and this where the start codon is, and we have 2 different letter reports, and we see that on the test. What we do is cross out those.
Depends on the instructions. Do not automatically, please, instructions of each question very carefully. If I ask you to replicate this DNA, are you going to disregard that? No, no, no. So it depends on the instruction of the question, okay? But there are some questions that specifically tell you to look for the start codon. In that, you must start with, disregard this and start with this, okay?
Okay.
Yeah.
To look for the start codon, we disregard that part.
Exactly.
And if you say transcribe it, we use it too, but you just don't use it in your formula.
Translation, protein.
Translation.
Exactly, exactly.
Transcription, not
What she's saying, when you transcribe it, it's a good idea to transcribe the whole thing, like CCAUG. If you transcribe the whole thing, it's easy for you to look for the start codon, AUG.
Yeah.
So do disregard that.
Okay.
Okay. Yeah. Any other question before we start the new? Yes, David. TAC, exactly. Yeah. That is the sense strand. Yeah, yeah. On the questions on the test, I will tell you which one is the sense strand, very specifically. Okay? I will tell you. Okay? All right. And remember, you always go from 5 carbon to 3 carbon. All right?
So if you have a static curiosity on the test and it says 3 prime, 5 prime, you can do it the reverse.
The reverse, exactly. But I'm not going to do that on the test for you. It's always from left to right, okay? Easy.
We have to make sure.
Want us to know which side is the
Hydroxyl group? Yep. Exactly. Yep. All right, let's finish up this chapter, then we'll have maybe half an hour or more to review today, okay, instead of starting the new chapter. Mutation. tell me please if this statement is true or false. A permanent change in RNA is called mutation. True or false? A permanent change in RNA is called mutation. Okay. RNA is what? This is RNA. This is DNA. So if I change my RNA right here, there's a change in RNA right here. Would that be mutation? It must be in DNA. The master plan must be messed up. That is mutation. Because if the copy is messed up and the original plan is fine, that's not mutation. Okay. With that? Well, I mean, the RNA is wrong. Yeah, you will make the wrong protein. But eventually, it is fixed, you see. The original master plan is fine. So if somehow this mRNA is messed up, maybe temporarily you will make wrong protein, but the original plan is fine. It is fixed. The permanent protein is safe. Okay? So maybe for the time being, let's say you're digesting food, okay? And the process of protein synthesis is how fast? 300 nucleotides, 300 amino acids in 20 seconds, okay? So maybe for 15, 20, 30 minutes or 2 or 3 hours, you are going to make the wrong enzyme to digest your food, may not be able to digest your food. But when you eat your food again, your mRNA is going to be duplicated again, so that will be fixed. Okay.
So, is there a diabetic medication that works in the mRNA?
There may be, David, I'm not sure about that. Yeah, there may be. Yeah, I am not aware of that. Yeah. Yeah. Do you know the name of that? Yeah, it is actually, yeah, diabetic medication. We will look up, yeah, we are going to talk about, yeah, there are several antibiotics. I'm not sure about diabetic medication, but there are several antibiotics that interfere with the synthesis of mRNA. Okay. Induce the synthesis of mRNA? Through a drug. I am not sure. I am not sure. You can speed up the process, let's say, if I understand it correctly, if you have, in human being, when you make insulin, okay, it takes very long time to produce insulin. You can take the gene from the DNA, from the human, put it in E. coli. In that way, you can induce the synthesis because E. coli grows much faster than human cells, right?
So that would be a culture.
Exactly. So you can produce large quantities of insulin in a petri dish, in a tub, much faster than human beings. That's what scientists have done. So yes, indirectly you can induce RNA synthesis. Yes. It is. It's actually practical use. That's how they make the insulin, large quantities of insulin. Bacteria, yes. Yeah. Yeah. Because the faster rate. All right, so mutation, a permanent heritable change in DNA. Heritable meaning that can be transferred from one generation to another, from one bacteria to another. That is mutation. Okay. Causes, no reason. When you grow old, you will see abnormal spots on your body. You may go to the sun, you may not go to the sun, right? So nature makes mistakes, and that nature's mistakes, they are called spontaneous mutations. A random change in DNA without any mutagen. Mutagen is anything that messes up the DNA, okay? Or you can say cancer-causing agent, mutagen, or oncogenic, okay? Mutagen, for example, if you smoke, nicotine is mutagen. You pump gas in your car, that's a mutagen. Asbestos, mutagen. UV rays, mutagen. So any physical and chemical agent that changes the DNA, spontaneous. So I told you if you drink water, it has trihalomethane in it, right? Mutagen. Okay. So Just by living today, you are exposed to thousands of mutagen. That's why the cancer rate is so high. Yeah. In any developed country, the rate is so high because we use so many preservatives in our food. We use gamma rays to zap our strawberries and so on. So the more chemical and physical agent we use, of course, the more cancer there will be. Induced mutations. Mutation that takes place in the presence of a mutagen. Okay, increases the rate of spontaneous mutation. Normally, when bacteria, this number is true for bacteria, not for humans. Whenever you grow your E. coli or bacillus or any bacteria, whenever it reaches a population from 1 million to 1 billion, one bacteria is going to be different, okay, on your plate. That's a mutant, okay? What caused it? Nature's goof up. No mutagen, but if you expose that culture, bacterial culture to, let's say, UV light or chemical, formaldehyde, okay, it is going to change or increase the rate. You may have 5 in 1 million to 5 in 5 billion. So that will be induced mutation. Induced mutation. If you live in a red brick home, old red brick home, old red bricks, they have large quantities of mercury in them, and mercury and lead. Old bricks. Okay, so your chances of getting cancer is greater if you live in an old castle that was made with red bricks. Learned that from one of my workshops. Types of mutations. Okay, are all mutations bad for the cell? Lethal? No, not really. Some mutations, they really have no bad effect on the cell. For example, the very first example. It's called point mutation, base substitution. That's the key word here. You do not add or delete. You substitute one nucleotide. Let's say this is DNA. 1C at nucleotide position number 6. You take out C and you put G. That is. But what if I take out a C and don't put anything? That's a different type of mutation. That's called frame shift. Like you learn in lab of a thymine dimer, right? When DNA is exposed to UV light, it makes thymine dimer. 2 T's are connected to each other. So that is an example of another type of mutation called frame shift. So sometime, let's say, if AAU become AAC, that's an example of neutral mutation, because both AAU and AAC, they make the same amino acid. Pardon me? Yeah, neutral mutation. It's also called point mutation. So point mutation, base substitution may lead to a neutral mutation. David? Number of nucleotides remains same. Number of nucleotides in point mutation or base substitution remains the same. In frame shift mutation, pardon me? Yeah, in this case, yes, redundancy. Yeah, like for example, for leucine, how many nucleotides do we have? How many codons we have? 6. So any one of the 6 can make the leucine. Okay.
And it's still considered a mutation.
It is a mutation, but that's why I said not all mutations are bad mutations. It's a mutation, but it's not bad. Okay.
Change is mutation means change.
Change in the DNA. Change in the DNA. Yeah. The outcome is still the same. Yeah. Yeah. I'm not sure what happened. Oh, this is normal DNA. You have, this is the normal sequence strand of DNA, methionine, lysine, phenylalanine, glycine, and stop. Whoops. Okay. This one we already talked about that. Done. In this case, AAU becomes AAG. Now you have a different amino acid. Instead of asparagine, you have lysine. Would cell be affected badly or not? How would cell be affected? Negatively or not? Depends on what type of protein this amino acid is making. Okay? If let's say this asparagine this mutation is affecting a protein that is involved in aerobic respiration, the cell will be affected adversely. The cell will not be able to make ATP. What if this protein is just making gelatinase right here? This part of the enzyme gelatinase. Will cell be able to live? Of course, but it will not be able to digest gelatinase. So it depends what type of protein is affected. It may or may not be harmful for the cell.
So then that's not neutral.
That's not neutral. No, because you're substituting now. Okay? Yeah. Third is definitely harmful. Okay. In humans, okay. In order to make normal hemoglobin A, you need GAG, glutamic acid. But GAG becomes GUG. I should have turned U red, not G red. It makes valine that changes the shape of the hemoglobin, crescent shape. Okay. So the substitution in the normal strand, okay, methionine, lysine, phenylalanine, we have a different amino acid right here. That is missense or point mutation. Nonsense mutation. Nonsense mutation. Normally in a gene, you have the stop codon where? At the end of the gene. But a nonsense mutation causes the formation of a stop somewhere in the middle of the gene or at the beginning of the gene. Okay. Look at this one. UAA is one of the stop codons, but a mutation. Now this mutation may be because of point mutation, because of frameshift mutation. Any mutation that leads to the formation of a stop codon somewhere in the middle of the gene. In this case, no protein. You have a single amino acid right here. So a nonsense mutation leads to the synthesis of a stop codon somewhere in the middle of the gene. Incomplete protein is made. Third type is the mutation that you have to do on the test. Yep. Frameshift mutation. Okay. This is the mutation in which you add or delete one or more than one nucleotides. This example shows you that the A is being deleted. Okay. So one strand of DNA is going to be fine, the other one is going to be affected, and you have a completely different protein, frameshift mutation. Okay. So let's practice this one. Okay. Go ahead, take your papers out, let's practice this one right here. You have this sense strand of DNA right here. At nucleotide number 6. Okay. Now please remember, you have to start with 1, 2, 3, 4, 5, 6. Okay? Yep. At nucleotide number 6, you are adding 2 new T's. 2 thymines are added. At nucleotide number 6, 1, 2, 3, 4, 5, 6. Yes.
Okay.
At nucleotide number 6, you are adding 2 new thymines. That's a frameshift mutation. How would that change this protein? First thing, you need to make your new genetic codes, right? And then make your mRNA, and then you translate it. Okay? Okay. What number did I say? 6. 1, 2, 3, 4, 5, 6. So you put new two T's right here. Okay. So the first codon will be fine. First codon, okay. CC, discard, okay. And the new code will be what? CAA. And then this, and then this. CCGGGU. And then you just look up these in your codon chart. AUT is MET. What is CAA? Anybody has chart handy? CAA? CAA is what? glycine, GLN, all right, GLN, CCG, CCG, proline, okay, pro, GUG, GGU, gly again, okay, oh, glycine, all right, and you disregard this right here, all right. So that's your frameshift mutation and the new protein, okay. Any question on mutation before we go to the last topic of this chapter, which is DNA transfer? Okay. Let's finish this up, then we'll come back and review anything you want, if anything you wish to. Come on. Move. Okay, fine. Sure, go ahead.
Like in sense, if we are disregarding the first two, G and G,
Yeah.
why is there counting these
Because if the question says to start with the start codon, then you have to start with the start codon. Because yeah, it depends on the question. Okay. Yes, Desiree? You start with GG. GG, exactly.
but only mutation
The question will, yeah. Oh, true. The reason we disregarded it because the instructions told you to do so. Not if the instruction doesn't tell you, then you start right from here.
This is for but the mutation.
Mutation.
Transcription.
I'm not sure. Repeat it again.
Like to disregard the first two, this will be, if the question says,
Yeah.
we will disregard it, but if it doesn't say,
You have to make your protein from the beginning. Okay.
You'll tell us, like if you're saying there's a mutation at nucleotide number 6.
Yes. Yeah.
If you say it just like that,
Mm-hmm.
then we have to start from the beginning.
It will tell you several things. Number one, it will tell you what type of mutation. Number two, where the mutation is taking place. And number three, if you should start with the start codon or not start with the start codon. It will tell you, the question will tell you all those things. Yep. What if I don't give you the start codon? That's very possible. I may not give you the start codon at all. Okay. Okay. If there is no start codon and you have, okay, GAC here, and I have dot, 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 dot here. What are you going to do? Not answer the question? Okay, there is no start codon. So it means, yeah.
So on this part of the test, it's just
Yeah.
a matter of the position that you said, binding,
Exactly. Yeah.
transcribe, or
What if I don't have start codon and I ask you to mutate this DNA now? Okay. At nucleotide position number 3, after 3, at nucleotide number 3, you delete a nucleotide from carbon position number 3. You delete it. C out, you make your new DNA. GAG. So it depends on the question. It could be different ways. Okay.
So that would
Yeah.
be your adding, your deleting.
Deleting. It could be, yeah.
Substitute, you'll also say like substitute for X position number.
Yep, I have to. I have to tell
Can
you.
you give us an example, like what you were doing?
Substitution, sure. Yeah, you can just take C out, and put G in.
So you will say like substitute at
Carbon position and nucleotide 3. Yeah.
Nucleotide 3, substitute G.
Yep. C is substituted with
You
G.
only substitute one nucleotide.
Correct.
You wouldn't say substitute GC or something like that.
I may.
Oh.
I may. It doesn't matter. Jim. Yeah. Go ahead.
Whatever letter we have.
Pardon me? On the test, there are some questions that where you have start and stop codon, others do not. Exactly. So it depends on the instructions. As long as you know what letter goes with what, you're in good shape. In DNA, for example, as long as you know this key right here, DNA, A goes with T, T with A, G with C, and C with G. That's your DNA. And in your RNA, if you know this principle, you don't have to worry about anything else. A goes with U, U goes with A, G, C, C, G. This is the template that you need to remember. Yes? On the test, I have actually eliminated these numbers just to confuse. If you want, I can put them on the test for you, 3' and 5', but always go from left to right. That's very simple, left to right. When you are doing transcription or translation or replication, left to right. Yeah. If you want, I could put 5' and 3' on the test for you. Okay. Yes. Absolutely. Absolutely. Yeah. All right. Last topic of chapter 8 is transfer of genetic material. Okay. How do bacteria transfer or acquire new DNA from other bacteria? Now you have learned one of the 3 methods, okay? Conjugation, okay? The other 2 that we have not talked about, okay? Transformation, okay? There are so many words that starts with T: transformation, transduction, translation, transcription. So you have to keep your T's straight, okay? Otherwise, it could be confusing. Transformation. First of all, you need only one metabolically active bacterium. You need one metabolically active bacterium for this process to take place. What else? Fragments of DNA, naked DNA. One bacteria, which is metabolically active, and fragments of DNA from other dead bacteria. For example, now this BC lake right here, let's say there are 2 bacteria living there, okay, in the lake, okay. One bacterium is still metabolically active, right here. And let's make this bacterium penicillin sensitive. This bacterium that is still metabolically active is penicillin sensitive, and its neighbor just died. Here's the bacterium. It died, okay, and it's broken down, cell wall is broken down. Now DNA is broken down into little pieces. Before this bacteria died, okay, it was penicillin resistant. It was penicillin resistant. Piece of DNA right here, XYZ, is floating in the cytoplasm or in the water, not in the cytoplasm, in the water. This bacterium is still metabolically active, right? Okay. This piece of DNA, XYZ, ends up in the cytoplasm of this bacteria. Accidentally, they don't seek out, I have to pick up this DNA or that DNA. So a process in which a metabolically active bacterium, one metabolically active bacterium, picks up a fragment of naked DNA from its environment. That is transformation. Now what will happen to this bacteria? Would it become what? Penicillin resistant, right here. Not seeking it. Like you take Tylenol for headache. How does it know to go to head? No, seriously, yeah. No, it's just diffusing all throughout, and it just randomly goes equal amount into all the cells. That's it.
I just had a hard time wrapping my hands around something dead becoming alive.
Let me show you. There was a scientist in Maryland by the name of Griffith. Okay, he conducted a very simple experiment to prove that this actually indeed does take place. This is called Griffith's experiment. Okay, let me first show it to you. Definition. Mode of genetic transfer in which DNA, fragment DNA from one bacterial cell, which is the donor, right here the donor, is picked up by the recipient. Now this says and subsequently undergoes recombination. Well, it may. There are two possibilities. Piece of DNA which may become part of the host. That is recombination, like that. Or it may not become part of the host chromosome, but it may become plasmid. Either way, the bacteria has new characteristic. It is resistant. So there are two possibilities. Although it says it becomes recombination. Recombination may take place, may not take place. Okay. Now let's look at Griffith's experiment. This is what I have on the board. This is in your book. Metabolically active bacterium, naked DNA, ends up in the cytoplasm. Okay. This one shows recombination right here. It may become recombinant, it may not. But now the cell is now genetically transformed. It has gained new characteristics. Yes, Tracy. It may be, okay, in this case I have resistance, but it can gain a whole lot of new characteristics. For example, in Griffith's experiment, this bacterium that was dead, it has capsule. And this bacterium did not have capsule. So this piece of DNA had the information to make capsule. So this bacteria now made the capsule. Depend on the coding. Absolutely. Yeah. All the time it happens in nature. Yeah. They have? Yeah. Oh, absolutely. The lifespan of each DNA is different. If you watch CSI or another, or programs really, like this, yeah. And they tell you the lifespan of DNA. DNA is a pretty tough structure. As compared to cell membrane, cell wall, they can survive for a very long period of time under right circumstances. Sandy?
So this is the process of how more and more bacteria is becoming
resistant. Exactly. That's one method. Now remember, in chapter 4 when we talk about conjugation, conjugation only happens in gram negative. But this, both gram positive and negative bacteria, they can acquire new characteristics.
Plasmids.
Exactly. They can get plasmid from other the bacteria. Tracy, yeah. Absolutely. Absolutely. Very good. Exactly. Oh, yes. Absolutely. Yeah. Yeah. Oh, that's interesting point. You see, this bacteria, let's say, is infected by a virus. Okay? A virus A. Another virus A cannot infect this bacteria, but a virus B can infect. Now you have two DNAs of the viruses in the same cell. You may have a mutant coming out from this cell. Sure. Yeah. Exactly. Yeah. And cold virus is a good example. That's why we don't have a good vaccine for cold, because it mutates so fast. Yeah. Yeah. Okay, here's Griffith's experiment right here. Griffith, he used Streptococcus pneumoniae. There are two different types of Streptococcus pneumoniae. Streptococcus pneumoniae. Okay. One is non-capsulated, uncapsulated, non-capsulated, like this. The other one is capsulated. Remember, capsule makes bacteria more pathogenic or resistant. And this is the DNA of this bacteria. and they have a capsule like this. Okay. Capsule. So in the first step of his experiment, what he did, he took capsulated bacteria, Streptococcus pneumoniae, and injected in a set of lab animals. What happened? Non-capsulated. No capsule, non-pathogenic, healthy animal. Then he took the capsulated bacteria, these bacteria right here, and heated them. Put it in a test tube, and let's put a test tube like this. And he heated it to get rid of the capsule. Remember, capsules are very, very sensitive structures. So when he heated them, capsules were gone. Cell wall was broken, okay, and DNA broke down into small pieces. DNA. So you have, this is your heat-killed suspension. Heat-killed culture. There are no metabolically active bacteria here. No metabolically active bacteria. So he took this suspension fragments and injected them into the third set of animals. What happened to them? There are no metabolically active bacteria, so they remain healthy. Then he took these non-capsulated bacteria and put them in this culture. Metabolically active bacteria. Okay. And what do you think happened to these? They picked up the DNA from the environment. What was that DNA for? Exactly, very good. So they became teenage mutant ninja bacteria now. Okay, like this, capsulated. They grew capsule. And when he injected this suspension in animals, what happened? Last one, they died. So this proves that metabolically active bacteria indeed pick up pieces of DNA from their environment.
That's only going to happen if you break it apart and the DNA is in fragments. You can't
Bacteria die in environment all the time. Yeah. So that happens all the time. Yeah.
Obviously, if you mix, I know this probably sounds stupid, but if you mix up encapsulated with non-encapsulated, unless you break that bond into a fragment, it's not going to...
Unlikely. Very unlikely.
Having them next to each other won't.
Unless they have a pilus or they have a carrier inside them, like a virus, which is the third method. Unlikely.
Right.
Unlikely. For this process, for the process of transformation to take place, you must have naked DNA. Yeah. Fragments. Okay. Yes, they can pick up.
Naked and fragment the same thing?
Same thing. Yeah. Well, there's another way DNA, this fragment DNA can end up in the virus. Viruses can pick up it into a new bacterium. That's a different process called transduction. But before we do that, let's look at conjugation, what we did in lab. Okay. Conjugation only takes place in gram-negative bacteria. So only gram-negative bacteria can benefit from this process, in which one bacterium, which is F-positive, well, two types of males actually, right? Two types of male, F-positive, and what is the other type of male that we used in lab? HFR. Yeah, I'll show you the diagram. So it could be F-positive or HFR, gives a copy of the DNA to the female through pilus. Physical connection is the pilus. Oops. All right. The first one you already learned in chapter 4. You have male bacteria and a female bacteria. Both are gram-negative. A copy is given to the female, male bacteria. That's it. This is mu from here to here. Some F factor, right here, instead of A, if the DNA is here or here, this bacteria is capable of making what? Pilus. This pink DNA has the information to grow pilus. So it doesn't matter if it is here or here. But the outcome is different when the F positive conjugates with the female, because this is a small piece of DNA, maybe 100 genes right here. It can be given to the female very quickly. Okay? But when they become one, how many genes do we have now? 1,100 genes right here. Integrated at the end of the chromosome, incorporated itself at the end of the chromosome. You see, remember the name of the amino acid that were at the beginning of the gene? Threonine and leucine. Okay, so female received threonine and leucine from the male. Okay, but she will never receive the tail end. Never, because it's so long, not enough time for the male to give all these genes to the female. So you can see right here, female did receive this light color blue DNA from the male, okay, but she never received the pink one. So when you conjugate HFR with F negative, what is the outcome? HFR and F negative. But F negative is not the original F negative, right? She has received a portion of DNA from the male, but not the F plasmid. Make sense? All right. Last method of DNA transfer is, this is the picture in your book, F positive, F negative, pilus, okay, yeah. Transduction. For this process to take place, you need 2 metabolically active bacteria and a virus. So I'm going to draw 3 bacteria. Bacterium A, it has this black DNA. Bacterium B, blue DNA. And bacterium C, with green DNA, or let's say red DNA, fine, red DNA. So there are 2 different bacteria. Okay. Now you need a virus. Virus is going to go to the first bacterium, could be any, ABC, could be any, doesn't have to be this one. Goes to the first bacterium, and the color of the DNA of the virus, a virus that infects bacterium, is called bacteriophage or phage or phage. Bacteriophage or phage. Or simply, if you hear the word phage, that's a bacterial virus, a phage. So DNA is going to go in. By the way, when a bacterial virus infects bacteria, the whole virus doesn't go in, only the DNA. Why? Rigid cell wall. Okay, bacteria have a rigid cell wall, so it prevents the penetration of the virus. Virus doesn't go in, only the DNA. How about humans? Do we have cell wall? Nope. So when HIV infects the T4 cell, the whole virus goes inside the cell, the head and everything. But here, we'll talk about this in chapter 13, viruses. But when a bacterial virus infects bacteria, only the nucleic acid, the DNA, goes inside the bacteria because of the rigid cell wall. Okay? Once the virus is inside, in about 20 to 30 minutes, okay, how many viruses are going to be in here? By the way, okay, there are 2 possibilities. We are talking about transduction, so I'll explain you this process. This is what's happening in Magic Johnson. This is HIV. This is T4. The virus is not multiplying in its T4 cells because it has become part of the T4 cells. So the cell is not going to die. If this happens, the cell doesn't die, the host doesn't die. Okay? What happens is, so whenever a virus infects a cell, a host cell, there are two possibilities. This is recombination. Cell is safe. Okay? But we are talking about transduction, so we'll do this. Remember the color of the bacterial DNA and the viral DNA. So virus starts to multiply like this. About 300 new viruses will come out from this one single host cell. 300. And some of these viruses, accidentally, they will pick up wrong DNA. Whose DNA? Bacterial DNA. Now let me ask you, is this a true virus? No, it's a virus look-alike now. Looks like a virus, but whose DNA is it carrying? Bacterial. So this virus, let's say it goes and it goes to bacterium B. What is it actually doing? Transferring the bacterial DNA from bacterium A to bacterium B. Okay. Now let me ask you this. Would it be safe to say that the virus has committed suicide? Yes, because virus cannot multiply. It's not viral DNA. It's bacterial DNA. So those viruses that accidentally pick up bacterial DNA, they have committed suicide. They cannot multiply. They're just like carriers. They transfer DNA and then they die. This is bacterial DNA. This may be the gene for what? A toxin, capsule, resistance. Okay? So this is transduction. How about this one right here? The virus, green one, goes to C. What is this? Transduction? No. First step, infection. This is infection. Infection. So this is transduction from A to B, transduction. And A to C is infection. Infection. Yep. Mm-hmm. Yep. Mm-hmm. Each virus is specific. Each virus. I'm not sure, Tracy, what other example? I'm sorry, say that again. In real life, scientists develop a virus in lab. Scientists, there's a man-made virus. Okay, this is exactly it looks like. Okay, like a polio virus. Okay, like this. It's an empty virus. Okay, this is the therapy for diseases for the future, gene therapy, because viruses are so specific. Let's say hepatitis virus. Where does it go? Liver. Okay, encephalitis virus to the brain. Okay, now if you want to fix the gene, let's say may have brain cancer, and you want to repair the gene that is causing the trouble in the brain, they will take this virus, encephalitis virus, okay, and they will put the fixed gene, the correct gene of the human inside this virus, inject it, and where do you think this virus is going to go? To the brain. They can fix any problem in the future by putting the right gene in it. Okay? Cardiac muscles is fixed, arthritis is fixed, whatever. That is the gene therapy. Okay? Virus as a carrier, area carrier to carry the right gene. Okay? That's the problem. They are playing God, so they don't know the outcomes of this. Whenever you have a man-made thing, they think they know this virus what's going to do, but you never know. Yeah. Okay. Yep. Another way you can do this is you can take hepatitis virus, pull out its DNA, and put the right gene in.
How do they pull out DNA?
Okay. Have you seen in vitro fertilization? In vitro fertilization? Yes, that's exactly how they do it. Yeah. Very carefully, I would say. Yeah. Okay. Jackie, did you have a question?
Why it's called infection? Because it's not the bacterial DNA.
Exactly. It's called infection because the virus is carrying its DNA.
Okay.
Its DNA. Jim?
Bacteria is going to die.
Absolutely. As soon as the virus starts to multiply, the cell is doomed. From A to C? It depends. It depends. From A to C, it may be recombination here. Not necessarily. It depends. When the viral DNA goes in, it may become part of the red DNA. Safe. Absolutely. Suicide. Absolutely. Absolutely. Jim?
You know, a fair question, why does it make bacteria phages? Why do people put it inside of eggs?
Whoa, okay. Viruses, they go inside the bacteria because viruses, they don't have any metabolism of their own. That's why they need a living host to grow. That's why viruses go inside the cell. They need another living cell so they can multiply. How did you create this? Okay. Let me just very quickly, may I borrow someone's book? I didn't bring my book here to answer what happened here. May I, please? Doesn't matter. It's okay. It's okay. Thank you. Okay. Okay, here we go. Now, how do viruses pick up this wrong DNA? Why? How? Okay, this is a good diagram right here. Not all of it, exactly, right here. Here we go. Here is a diagram, okay, that shows you what happens when a virus, this is a bacteriophage, looks like a spaceship, beautiful spaceship, really. And look at the color of the DNA here. Oh, that's not a good diagram, sorry. Okay. But look at this virus injects its nucleic acid into this bacteria, right? Okay. Now, the multiplication of virus is not like bacteria, one virus to another virus, 3, 4, 5. It's more like constructing a car. When you build a car, the tires are made somewhere else, the windshield is made somewhere else, and then the engine is made somewhere else, right? So when a virus DNA goes in here, see what happens here, right here. The different parts of the viruses are made separately, the tail, the head, the pins, and whatever. And then there's an assembly line. Just like when you build a car, you have assembly, the tires are fit together, okay? The engine is put in, right? Look what happens here now. Here's the DNA. Here's the tail pin and all. This is called assembly. And the mistakes are made during assembly. You wanted to put the 4 tires, but you put the VW tire on the car. Okay, really? So during assembly, or this is called maturation, viruses pick up wrong DNA. Instead of their own DNA, they pick up bacterial DNA. Okay? That's why the mistakes are made during maturation or assembly. Okay? Yep. Exactly, because it picks up the DNA from different viruses and hosts and all that, so it's a completely different strain. Okay. Jim, does this explain why the viruses pick up wrong DNA? Lindsay? Yep. Okay. Now let me show you. Okay. Go go into the detail, but here we are. This is a very nice diagram that shows you two possibilities. When a virus goes in a cell, you see right here, this is the one that we just talked about. Goes in, starts to multiply, and what happens? The host cell dies. Look at this one right here, Magic Johnson, right here. This DNA becomes part of the host DNA. What happened to the cell? No death. So whenever a virus infects a cell, a host, there are two possibilities. It may go into lytic cycle or death cycle, or it may go into lysogenic cycle where there is no death. The cell remains alive. Yep. If it goes in lytic cycle, silent symptoms right away. But no one knows what causes this virus to start to multiply in some host and not the other. Okay. Right here, this is carrier. This is carrier phase. The person has it, but asymptomatic. Yes, it can. It can. No one knows. Yeah. Some people, they start to show signs and symptoms within few months. Others, they may take years, and sometimes they may not. They do not know. If someone knows what, billionaire overnight. Yep. Okay. Exactly. Yeah. Yep. Yeah. Yep. Exactly. Absolutely. That's why it remains dormant in your body. Exactly. 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 Yes. Yeah. Shingles. Yeah. Chickenpox, shingles remains dormant in your body, and you get shingles like that. Yeah. Yeah. I'm not really sure about that. Yeah, I don't know. Yeah. Yeah, I'm not sure. Yeah. Yeah. Shingle, they say stress. I don't know. I don't know. I have none. Okay. Go ahead. Of the host. Yes, absolutely. No one knows why, how. Yep. It may become part of the host, and within a few months it may separate and start into lytic cycle. Okay. Yeah. So this is the diagram in your book about transduction. I like mine better. Look at the colors here. Pink, magenta color, Prof. Yeah, this is bacteriophage. Goes in, starts to multiply. You have two different colors of viruses coming out, right? Purple one is bacterial DNA. This one they show recombination, but doesn't happen all the time. It may become recombinant or die. The end. Chapter number 8. How much time do we have? 15 minutes, okay. Huh? A lot of time. Fawzia, go ahead. Thank you. Yes, yes. Questions, please, from any chapter. 5, 6, 7, 8. Jim. Conjugation? Correct. Yep, correct. Yep. Conjugation can only take place in gram-negative bacteria. Yep. Okay. Look at your pretest and tell me if there's anything you want me to go over from the pretest, please. Or any topic. Yes.
Video number 15 yesterday.
Okay.
And it was saying that in the last LAG phase,
Yes.
it's the cell duplicate.
No. Cells do not duplicate, cell components duplicate. Cell components duplicate in LAG phase. Cells multiply in LOG phase. Yep.
No, it's
Yep.
it's
Yeah. Cell components, SEMPAR, DNA, ribosome, cytoplasm. They do. Did you have a question? Yeah.
that was DNA duplicate
Yeah.
in the lag stage.
Yeah.
In the log stage is when the cells duplicate. We're doing the glycolysis
Glycolysis, okay.
and Krebs cycle.
Yep.
When you asked about how many, the end products,
Sure.
there was a mention, are you going to be specific when it says like 8 NADH is at the end of the Krebs cycle?
Correct.
Break it
Yeah.
up, there's 6 from,
Yeah. Not going to be that specific. Just remember there are 8. Okay, 8 at the end of Krebs cycle. Yeah.
Not how many are used.
No, just as long as you memorize the end products of all 3 phases, you're in good shape. 2 ATPs net, 4 total. Right? 2 NADHs, 2 pyruvic acid. End product of glycolysis. Krebs cycle, 2 ATPs, net and total same, 2 FADHs, 8 NADHs, and 4 CO2. And then 34 ATPs and water. That's good.
water in general.
Yep. Yep. 34 ATPs, water. Electron transport chain. Krebs cycle. Glycolysis. Okay. How about fermentation? This is aerobic. Fermentation. That's it. 2 ATPs, 2 NADHs, and then remember glycolysis only, so you have pyruvic acid, 2 pyruvic acid, which can be converted into acid, alcohol, or both. That's your fermentation. Acid, alcohol, or both. Right? Acid, alcohol, or both. Which one will make the most ATP here? The one bacteria that's making acid, or the yeast, or E. coli? Okay, which bacterium will make the most ATP? Lactobacillus bulgaricus, yeast, or E. coli? Which of these 3 will make the most ATP? The same. Because they're all going through glycolysis only. There is no other place to make ATP. Same number of ATP.
So you're going to give an example like of 2 different bacteria, one that would be...
Maybe. Yeah.
Question mark. CD.
Only if I told you. Only 5, yeah. It is important to know all the names, you know that, okay? Yeah, so I go.
To make sure we know which are aerobic and anaerobic going into.
Sure, yes, 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 yeah. Good day? Okay. Oh, you have 5 minutes, sorry. I know, you don't want to leave today. You don't want to. Go ahead, 5 minutes we have, all right? Yes, questions? How about Opron? Everybody okay with Opron?
No, not at all.
Okay, who's going to take a step forward and explain Opron to the class? Oh, come on.
I'll take it.
All right. Quickly, let's compare the two operons. Operon, what is it? A string of genes, set of genes, regulator gene, promoter gene, operator gene, and structural genes. What do? They control the synthesis of proteins that are not made all the time. That's what the operon does. Okay, control proteins that are not made all the time. So you have two types of operon, inducible and repressible. Repressible. What's the difference? In inducible operon, when the repressor is made, the repressor protein is made, it is active. And in repressible operon, inactive repressor. Inactive repressor protein. Active means bind to the operator. So when you make the repressor, this is active repressor, this is inactive repressor. Active repressor goes to the operator. Let me ask you this. Active repressor binds to the operator only in inducible operon, true or false? Okay. Active repressor in general always binds to the operator. True. Always. You see, this is inactive, that's why it cannot bind, but once it becomes active, where does it go? Operator. So active repressor in both operons, it binds to the operator. So that statement is true. Active repressor in both operons binds to the operator. Okay? And what's the function of active repressor? To block the passage of RNA polymerase. That's the function of active repressor. RNA polymerase. It blocks the passage, cannot go forward, no transcription, no translation, no protein. Okay? But remember, in repressible, because it is inactive, you are going to make the protein, right, at the beginning. In inducible operon, it is active, so you are not going to make the protein. Opposite. He is here. Okay, good day. Right. All right. Pardon me? Yes. Yeah. Yeah. Saccharomyces cerevisiae. Yeah. Aerobic micrococcus luteus only, I think that's the, I gave you two examples under aerobic when we were talking about chapter 6. It's in chapter 6, yeah. Yeah, aerobic micrococcus luteus and Pseudomonas aeruginosa. Those are